I want to welcome you all to the Fall 2025 Applied Ecology Minors Research Symposium. Thank you so much for joining us today. Thank you, Heather, for this amazing advertisement. I'm really excited to share that as of December 2025, we will have graduated 188 students with the Applied Ecology minor since we began. So that's pretty awesome. We're getting very close to 200. Before we get started, I want to acknowledge that the lands within and surrounding present-day Raleigh are the traditional homelands and gathering places of many Indigenous peoples, including 8 federally and state-recognized tribes: the Kawari, the Eastern Band of Cherokee, Haliwa-Saponi, Lumbee, Meherrin, Occaneechi Band of the Saponi Nation, the Saponi, and Waccamaw-Siouan. We share an ongoing responsibility to safeguard these lands and to respect the sovereignty of the tribes and Indigenous nations residing in North Carolina. Our goals in applied ecology are to study the world around us and apply what we learn to inform and empower stewardship. But it's important to remember that long before settler scientists decided that this was important, expert knowledge and care for the world around us has been applied as an integral part of Indigenous cultures in North Carolina and around the world. Acknowledgement is a form of decolonization, dismantling existing power structures. In order to change systems of oppression, we need to first acknowledge that the system is built on oppression. North Carolina State University is a land-grant university, which means it originally received funding through the Morrill Act, federal legislation that bought or forcibly stole lands from Indigenous tribes across what is now the United States. The sale of Indigenous lands originally raised $134,920 to fund NC State. That's the equivalent of $3,810,578 today. I hope that we can continue to disrupt and dismantle colonization beyond a simple territory acknowledgement by supporting and empowering each of you as scientists. So thank you for joining us so that we can celebrate ongoing students' research. With that, I'm really excited to learn from our speakers today. So here is our lineup. And first, we have Ivy Azzarita.
Hi, everyone. My name is Ivy Azzarita. This semester, I worked in the Seed Library in Kilgore Hall. And for my applied ecology research experience, I tested the effectiveness of smoke in overcoming seed dormancy. There we go. This is specifically relevant to the longleaf pine ecosystems that are here in North Carolina. They're also found along the Atlantic coast from Virginia through Texas. This ecosystem is characterized by the domination in the overstory of the longleaf pine, a lack of a mid-story layer, and a strong ground cover layer with lots of species diversity. They also are characterized by having lots of wildfires, which reduce competition between the plant species. These fires are able to accomplish this by reducing the upper canopy layer, allowing more sunlight to penetrate down to that ground cover layer, allowing those species to thrive. Because of this, many species have co-evolved with the smoke, which has allowed smoke to trigger germination in many of these species. For some background information on overcoming seed dormancy, I'm going to be talking about stratification a good bit in this presentation. This is the overcoming of physiological dormancy by putting seeds through a cool or warm period to overcome a chemical barrier to germination. There's also scarification, in which seeds need to overcome a physical barrier to germination, which is typically a hard outer seed coat, which needs to be broken to allow water to enter the embryo so the seed can germinate. And I also want to mention carotenoid. This is a natural hormone found in smoke, which can help many seed species overcome this physiological dormancy by signaling ideal growth conditions in this case, more sunlight. These are my hypothesis for this experiment is that grasses and herbaceous species would germinate better than woody species with the smoke. This is because in nature, smoke tends to benefit the lower growing species, which tend to be herbaceous or grass species. I also thought that species without existing cold stratification requirements would more easily overcome dormancy with smoke, as they would have less difficulty overcoming dormancy in general. Okay. These are my methods and materials. I completed this experiment in the lab in Marianne Fox. I began by soaking filter paper in DI water for control and a liquid smoke solution for my test. This was then split into 32 different petri dishes. Each of my 8 native species had 2 dishes in the control and 2 for test. These were then all parafilmed and placed under grow lights in the lab, which remained on for 16 hours a day. And then I observed seed germination and recorded their data and transplanted any seedlings as they germinated. The species I selected for this experiment were all native to North Carolina longleaf pine ecosystems. The ones that I specifically used in the experiment were based on seed availability. And I used a variety of woody, herbaceous, and grass species. I originally intended to test carnivorous species. However, I was unable to obtain seeds until much later in the germination, and I was not able to collect much of a data set to present today. These are my woody species. I have Colmia latifolia, Pinus virginiana, and Styrella racemiflora. I did want to mention that Styrella racemiflora does only have a 30% expected germination rate, as stated by the source of Sheffield seeds, as it does typically reproduce vegetatively. For my herbaceous species, I selected Lupinus perennis, subspecies perennis, Tephrosia virginiana, and Oenothera macrocarpa. And for my grasses, I have Andropogon virginicus and Schizocherium scoparium. I also wanted to take a moment to mention the liquid smoke I used. This is typically found in grocery stores and often used for cooking. However, it does contain the hormone of carotenoid. So I mixed this in a dilute solution of 1 to 50 DI water to test my species, as it is commonly able to be used in germination tests. I will now be going into my results. So this is my overall germination data. As you can see, every species kind of did its own thing. The pine germinated very quickly, whereas the Styrella did not germinate at all. So I'm going to break this down into smaller graphs based on different factors. Here's my data broken down by type of species. As you can see, the data points for each species are pretty close together. There was not much of a difference between the smoke, which are the lines with the circles, and the control, which are the ones with the diamonds. In some species, you can see that the smoke kind of jump-started the germination process. As with the Comia, the smoke started off a lot stronger than the control, but things evened out pretty quickly. For my grass species, the Schizocherium performed a good bit better with the smoke, but the data was pretty close together, and the Andropogon did not do as well with the smoke. It did better in the control. And interestingly enough, neither of these species reached their expected germination rates of 95 and 97%. And for the herbaceous species, they all generally reached around 40 germination. The Oenothera and the Tephrosia both perform slightly better with the smoke. However, once again, the data points are very close together. Here's this information again broken up by the species with longer stratification periods, so 30 days or greater, and shorter stratification periods of 10 days or no stratification period. And again, there was not a significant difference between the germination and the short and the long stratification periods and there's not much of a difference between the smoke and the control in each of these. However, I did notice that the species with the shorter germination periods had a few more species that performed better with the smoke. My biggest challenge in this experiment was mold. There was an infection that quickly spread across all of my petri dishes. This was seen throughout the lab, not just in my experiment. We're not entirely sure where the problem came from. I did regularly treat my seeds that were infected with a 20% bleach solution to try to kill mold spores while not harming the seeds. However, some seeds did rot before they were able to germinate, which did impact my data. One interesting thing I did notice, though, is I tended to see a bit more mold on the smoke petri dishes, which is not what I was testing, but still an interesting observation. And then I finalized my experiment by cutting the remaining seeds of my Cyrilla and my herbaceous species to test if they had any embryos. If the seed did not have an embryo, it would not have been able to germinate. I did not test the pine because they had all germinated, and my grass and colmia seeds were too small to really see an embryo even under a microscope. For my Cyrilla, 14% of the seeds in the control had embryos and 10% in the test had embryos, indicating that not many were actually able to germinate. And for my herbaceous species, the Tephrosia and the lupine, all of the remaining seeds did have embryos, but the Oenothera, only 80% of the remaining seeds in the control had embryos, and only 70% in the test had embryos. To wrap things up, I would say that my hypothesis was proven to be largely inconclusive. While I did notice that some herbaceous and grass species did perform slightly better with smoke, as I mentioned, those data points were very close together, and I did not see much of a difference between germination rates in the control and in the smoke. This indicates that the smoke did not prove to have a strong effect on any of these particular species that I tested, indicating that there was very little reaction to pyrokinolide. Because of this, it is still possible that they could germinate at their normal rates following a wildfire and compete with species that do require this hormone for germination as normal. Yeah, thank you very much. Thank you to everyone who helped me with my experiment, especially to Liz Meyer and my friends who showed up to support me today. I'll take any questions. Yes.
So I would imagine that depending what's being burned has different compounds in the smoke. Does it all contain pyrokinolide? What's that?
Pyrokinolide, as far as I'm aware, that is a hormone in the smoke itself. I did actually read someone's thesis that they tested different, burning different types of things to get different results, and I want to say all of their seeds were inconclusive. They didn't see much of an impact on any of them.
It seems to be a pretty common thing, all smokes.
Yeah.
But it might vary.
Mm-hmm.
So
Okay. Hi, my name is Lorelei. I'm a senior this year in anthropology, and this is the social network analysis of two lemur cata family groups at the Duke Lemur Center. It is a comparative study of female dominance and group behavioral dynamics in the captive ring-tailed lemurs that are housed there. Oh, hold on. Did I? No, it didn't work. Oh.
Yeah. You got it.
Okay. Okay. So lemurs are a primate species endemic to Madagascar and are commonly found in captive care due to ongoing threats in their natural habitats due to anthropogenic disturbance like deforestation and the legal pet trade, to name a few things. But what makes them so interesting is that female dominance, a rare social structure, has been observed to some extent in all lemur species, in which females are either completely socially dominant or at least not subordinate to their male counterparts. Because lemur group dynamics give us the earliest example of primate sociality, and their female dominant behavior generally separates them from every other primate group, lemurs act as an important and unique group to analyze and developing a further understanding of the evolution of male-female social relationships and general primate social structure. Female dominance within lemurs is thought to be an adaptation to the high cost of reproduction and a lack of resources compared to other primate species in their natural habitats. The lack of resources can be attributed to the variable and difficult terrain of Madagascar that makes vital resources a bit more difficult to come by. But female dominance has combated this struggle through giving females priority access to those resources that are vital to successful reproduction. Ring-tailed lemurs, or lemur cata, specifically have distinct male and female dominance hierarchies and have shown complete female dominance in both captive and wild environments. This dominance doesn't end in just dominance over males, but also alpha females are given priority over other females within the group. Although females have this distinct dominance over males, linear dominance hierarchies occur within each sex, where an alpha male typically also has priority access over other males to access to females in the wild, and then food in captivity and the wild. However, these alpha males still primarily lack any dominance over other females, no matter their age or relation to each other. Sorry. I cannot figure this out. There we go. Okay. This leads me to my main research objective through this project. I wanted to utilize behavioral observation and social network analysis to understand how agonistic, aversive, and affiliative interactions vary between two groups of captive ring-tailed lemurs and the individuals within those groups depending on their age, sex, and role within each group. With my findings, I hope to provide a better understanding of these specific groups of ring-tailed lemurs and lemur social behavior in general, while discovering how certain demographics can affect the initiated and received behaviors by specific actors. Additionally, information provided through this type of behavioral study can aid in the future of captive animal management by informing how to maintain a stable network through identifying the most important individuals. Yes, okay. It was hypothesized that in both groups, the matriarchal female will showcase more dominance behavior compared to subordinate females and males, exhibiting the highest centrality via initiating agonistic behaviors and receiving aversive and affiliative behaviors. Additionally, in comparing the two groups, I hypothesized that Group B will showcase more tension through larger group centralization in the agonistic and aversive behavioral categories due to the presence of infants and more females than Group A, and therefore Group A may appear to be a more stable network based on their affiliative network, as social structure is maintained primarily through affiliative behaviors and ring-tailed lemurs. Okay, so behavioral observations took place 3 days a week for 4 weeks at the Duke Lemur Center in Durham, North Carolina. This past summer, I accumulated around 48 hours total of observation, 24 hours per group. Behavior was observed regardless of their location that day, that specific day. So whether they were indoors due to rain or because they just didn't feel like going outside, or in their respective free-ranging forest enclosures. This is the summarized ethogram I formulated based on definitions by past research articles representing the common social behaviors among ring-tailed lemurs within each of those measured categories: agonistic aggressive, agonistic aversive, or submissive and affiliative behaviors. I also took note of any individual behaviors such as feeding or self-grooming or anything like that by oneself, but those were not included in the observation. Those observations were not included in my analysis to focus on interactions between two or more individuals. The behaviors were weighted basically by extremity levels specific to each behavioral category, but also as a way to pinpoint frequency efficiently in the master data set for all observations. Within each category, the weights were appointed to behaviors on a scale, with the highest number in each being the most extreme behavior in that category. I collected my observational data using the focal animal sampling method, wherein every instance of behavior exhibited by a randomly selected focal individual within the behavioral categories outlined by this ethogram was recorded during a 15-minute observation window per each member of the two lemur groups. And then all data points were recorded according to all approaches and leaves initiated by or directed at the focal animal being observed. So now I'm going to introduce the study subjects, the two family groups that I looked at. Throughout this presentation, the family groups will be referred to as Group A, or Elena's group, or Group B, Sophia's group. This is a table of Group A, led by matriarch Elena, her mating partner Stuart, and their three kids, Mr. Popper, Jasper, and Cyrilla. And then every picture that is featured here are either pictures that I took of the family groups that I looked at or provided of the family groups that I looked at by the Duke Lemur Center Instagram. And then this is a table of Group B, led by matriarch Sophia with her mating partner Randy and their daughters Nemesis, Nyx, and then their baby twins. During the time of this study, the twins were only 4 months old and weren't given names yet, but I was able to identify them. I gave them the IDs B1 and B2 for baby 1 and 2, and they have since been named Elizabeth and Doris. But for the purpose of this study, I did not focal them for behavioral observations because they were very young and they had total reliance on mom during the time of study. So I just don't think it would have worked out that very well. But I did note all interactions initiated by other members towards them. I did not include those in the specific analyses today, but I think that that could serve for some interesting analyses in the future on each individual's role in the care of the newborn infants. And then I would also like to note that the presence of these infants likely attributed to many of the behaviors and dynamics that were exhibited through my time observing them, as it is common for additional attention, even if unwanted, to be given to the mother and her infants by other group members following birth. It is also common for mothers to initiate more aggressive interactions towards other members following giving birth. Yes, okay. So starting with my methods, I want to explain social network analysis a little bit and the importance of social relationships when studying groups. Social relationships are vital to understanding individual and group well-being of many species living both in the wild and captivity, as the bonds those animals form are vital to their physical and psychological health. Understanding these relationships are extremely important in the care of captive animals specifically, though, because those animals have less choice over their social relationships. Social network analysis specifically has been this tool emerging to assess group social structure using different behavioral measures in both humans and various animal species. It can generally be defined as the use of matrix-based data to analyze individual, dyadic, or group-level social interactions through any networks-based analyses. Within the past few decades, social network analysis has really emerged in primatology, but studies like applying it to primate social networks and comparing them specifically are still rare. For this specific study, social network analysis was utilized through UCINet, a software, an accessible and free software to perform those analyses. And to quantify the certain metrics of ring-tailed lemur social dynamics, the metrics I will be talking about today include reciprocity, degree centrality, and group centralization. I was able, by going through the master data set of all of the observations, I was able to create adjacency matrices, which are these, to put into UCINet to test those specific metrics, each number being how many times the member in the left column initiated that type of interaction to the corresponding member in the top row. And then the numbers in parentheses showcase the total number of behaviors falling under each category exhibited by that group as a whole. Segueing more into the results, we can see that Group A has significantly more affiliative interactions than Group B, even though I observed them for a similar amount of time. But this can be attributed to the fact that Group B more often than not, especially compared to Group A, decided to stay indoors, which could be coinciding with a lack of energy and therefore a lack of social interaction. And then also to note, the agonistic aggressive behaviors in Group B are twice the size as the agonistic aggressive behaviors in Group A. Okay, so the results: reciprocity calculates the amount of ties or interactions that are reciprocated within a network and can be reflective of a group's overall stability. Although both groups showcase 100% reciprocity in affiliative behaviors, and both groups show 0% reciprocation in aversive behaviors, they differ in aggressive behavior reciprocation, referring to the matrices, with Group B having twice the amount of aggressive behavior than Group A. The reciprocation level indicates that 33.3% of aggressive interactions were reciprocated in Group B, and Group A being at 0%. These results do more or less support the hypothesis that Group A has better network stability than Group B through the presence of 100% affiliative reciprocation and Group B's higher percentage in aggressive reciprocation. Degree centrality can be used to identify which nodes hold more importance to their network based on the number of connections that they hold with other individuals. The out degree indicates the extent to which a node initiates a behavior towards another, while the in degree refers to the extent to which a node is the recipient of a behavior. And then group centralization is measured as a proportion on a scale of 0 to 1 and is an indicator of whether or not a group contains individuals who are more important than others in the group. A score closer to 0 shows that social interactions are more evenly distributed, and a score closer to 1 shows that there is a more central individual in that group. So we can see that Group A's aggressive out degree centralization compared to their in degree centralization is lower, indicating that there is a more central individual when it comes to receiving behavior compared to initiating it. In this case, I would say that Jasper is that more central individual when it comes to aggressive behaviors, which tracks with the expectation that the youngest male would be the most subordinate in that group. We can also note that Elena, the dominant female, has initiated the most aggressive behavior compared to anyone else at 5 initiations, which supports the hypothesis and tracks with the expectations of female dominance behavior. Group A's aversive out degree centralization compared to its in degree centralization is lower. This also indicates that once again, there is a more central individual in receiving the specific behavior. In this case, that individual is Elena, meaning she has received the most submissive behaviors by others, all by the male individuals in that group. Interesting though, we can also see that Cyrilla is starting to emerge in that female dominance because she has received a submissive behavior and has enacted an aggressive behavior. And then the group's affiliative centralization does back up the 100% reciprocation level, as both of their scores are lower, closer to 0, showing that affiliative interactions were more evenly distributed. And then Group B's agonistic aggressive centralization is much larger than its in degree. This can be supported by seeing that the only node that never received any aggressive behavior was Sophia, the dominant female, while Nyx really took the forefront with initiating aggressive behavior, likely being the cause for their higher out degree centralization, and she is the most centralized individual when it comes to initiating that behavior. Randy, the sole male, is the only individual who has not initiated any aggressive behavior and is the recipient of the most, tied with the eldest daughter, Nemesis. Their aversive behavior centralizations are both relatively high. Their centralization is reflective of Sophia solely receiving submissive behavior and Randy solely initiating it, both with more instances than both Nemesis and Nyx. Their affiliative centralizations are actually the same exact as Group A's, which also correlates to their 100% reciprocation. Okay, and then furthermore, I also created network visualizations through UCINet's NetDraw function. Here, the shapes represent the node's age group, with a circle being an adult and a square being a young adult. The colors showcase the sex of the node. line thickness is proportional to the tie strength or the number of interactions observed between dyads, with thicker lines indicating increased strength and thinner lines indicating weaker. Similarly, the direction of the arrowheads indicate the direction and size of the arrowheads indicate the presence and strength of the initiator versus receiver behavior. And then comparing the aggressive relationships of these two groups, we can see that these visualizations supports the degree centralization results, with group A having a clear most central individual in Jasper, and group B being more evenly distributed as everyone initiates and receives aggressive behavior besides Randy and Sophia. Also in group A, we can see that Cyrilla is kind of this outlier, with Jasper being most central. Network visualization additionally acts as a helpful tool in dealing with the complexities of social network data. And then comparing the aversive relationships of the two groups, we can clearly see that in both the males are initiating the most aversive behaviors, no matter their age, primarily to the dominant females, Elena and Sophia. This further supports the idea that sex has significant impact on behavior. And then we can clearly see here that the abundance of affiliative behavior initiations by Cyrilla, there is an abundance of affiliated behaviors by Cyrilla towards Jasper. This is important in specifying dyadic relationships within the larger network, because even if there is 100% reciprocation shown by both, and in this case, even when groups have the same degree centralization, some ties are bound to be stronger than others. These diagrams give a visual representation into the possible dynamics that are most important to the overall network stability, while also revealing expected dyadic dynamics. And then discussion. Overall, my results have confirmed previously established ring-tailed lemur group dynamics, specifically in instances regarding female dominance behavior, supporting the premise that agonistic interactions determine dominance, but also in the fact that males rarely show aggression due to its root in decreasing attractiveness to females. Utilizing social network analysis not only provides information provide information to institutions on the stability and the predictors of instability in networks, better informing general management decisions, but by identifying the most central individual in a network, caretakers can make beneficial inferences for training by selecting that individual that will be able to faster diffuse that information to the rest of the group, and concerning disease transmission, will know which individual should likely be isolated or treated first. In the broader world of applied ecology, trade-offs associated with group living can emerge at the individual, group, or population level, and exploring the interactions between individuals at each of these levels can help us to better understand the costs and benefits of these trade-offs. Additionally, the growing evidence in the field of primatology that the structure of a network is directly linked to its functioning furthers the idea that networks are selected in order to increase the fitness of its members. Therefore, studying lemur groups' social networks, even in captive settings, can inform larger questions and ideas in the field of primate behavioral ecology. The primary challenge I faced in conducting this research was learning how to use and understand UCINet as a new software. It was a bit difficult to navigate, but I think that with more experience with the software and by utilizing more of its metrics and going with more long-term data, this challenge can be faced. Yeah. I can't read my last part because it's hidden, but overall, relationships do need to be studied in the context in which they evolve as a part of a social group and over a more significant period of time. So my suggestion for future research would be to utilize long-term behavior on a larger scale of observations for more members and more groups to kind of better represent the complexities of primate social groups, and then also to reflect ongoing complex lived experiences and the evolution of those groups. I would like to thank the animal care team, research staff, and tour guides at the Duke Lemur Center for their support, trust, and with providing specific information about each of the individuals. And of course, I would like to thank my research mentor, Dr. Tara Clark, for guiding me through this entire independent research process and teaching me how to identify and understand ring-tailed lemur behavior. Your support has meant everything. And then questions. Sure.
So, can you remind me, is Sophia the mom and the matriarch?
Mhm.
So do you think that Nyx was like subbing in as alpha aggressor?
I was hoping someone would ask this. I think, yeah, Nyx and Nemesis's kind of like dynamic is really interesting because it doesn't necessarily reflect like typical ring-tailed lemur matriarchal like behavior, because typically the oldest is usually more dominant over the youngest. But I think just based off of like firsthand observing their behaviors and their personalities, I think that Nyx is kind of caught between like the two babies and the oldest daughter, and I think that she is kind of just trying to compete. And I think that it's just kind of a competition for those resources, but also for the attention by the mom and things like that. So like it was very evident in their personalities that Nemesis would be the more bullied one, and especially by Nyx.
Would you expect that group dynamic to like become more centralized as the babies grow up?
Probably. I think with this group specifically, I don't remember the exact details, but I know Nemesis's twin sister, I believe, was actually evicted from the group at one point because of her subordinates. So I definitely think that this group is very complicated in their dynamics and very interesting. And I do think those babies could possibly even reflect that pattern.
Right. One other question.
Yes.
And then I'll check the rest of the room. But so it really stood out to me that Jasper is bullied a lot, not to put value terms on it.
Yes.
Right. He's receiving a lot of aggression. He also gets so much affiliation. Like he's in a really strong dyad with Cyrilla. Do you think that that's really important to like compensate for that aggression?
Yes.
Like would a male ever leave the group?
I think, well, in the wild, males do typically, I can't remember
they
the
get
exact
kicked
terms.
out
Yeah,
when they're
they
sexually
leave.
mature.
Yeah.
But would he leave the group if he was receiving too much aggression?
I think that there's potential if it escalated to a certain amount, but I also think that it makes sense just with ring-tailed lemur patterns that he will receive that aggression no matter what, just kind of maintain that hierarchy. And I also think that male ring-tailed lemurs, they do kind of like it's in their nature to combat that aggressive behavior by initiating more affiliative interactions. So I think they kind of balance each other out.
Thanks.
Yeah, of course.
My curiosity is just does anybody else have any questions?
Yes.
Great presentation. I was wondering, you had mentioned that like one group spent more time with side, but wondering if you like reported that information, like looked into kind of like the effect that had.
Yeah. I took note every time. I just had like my little notebook. So my materials were my notebook and my eyeballs, really. So I took note every single time I went on whether or not those ringtails were going to be free ranging that day. I haven't specifically put that into my broader analysis, but I think, and I've thought about the further implications of that. And there's like so many other like little factors that I think that could explain specific behavior that I would like to look into at a later point, possibly.
Thank you again, Lorelei.
Hi, everybody. My name is Shea Hoffacker. Thank you for coming to the Applied Ecology Minors Research Symposium. Today, I'll be telling you about the study I designed for my internal learning experience, where I took a look at bluebird nesting preferences and nesting success in response to pallet height. I would first like to thank the village of people who supported me, including my mentor, Alexio Maneshi, Iris and Randy Sensing, the NCSU data science support staff, specifically Faye Yao, and as well as Dr. Erin McKenney, who provided me with countless feedback. Eastern bluebird populations began declining throughout the late 1800s to 20th century, and were at an all-time low in the 1960s and 1970s. This graph shows data from the Audubon Society Christmas bird count from 1941 to 2003 in the United States. Bluebird population decline was mainly due to habitat loss of open space, as well as introduction of invasive species, such as the house sparrow and European starling. both of which aggressively compete for the same nesting cavities as bluebirds and are known for destroying bluebird eggs, killing adult bluebirds, and evicting bluebirds from their nests. Bluebirds also have a low nesting success rate, about 60%, which is a lot lower compared to other cavity nesting species. Studies have also been done showing that 70% of bluebirds die before reaching their first birthday. Though this is not as significant of a factor, this could be another reason for their decline. Population size began increasing steadily, about 2% each year, when conservation efforts like installing nest boxes were put into practice. This study used 30 NAB-style nest boxes on Lonnie Pool Golf Course, which look like this image on the left. On the right is a schematic for the NAB's nest box style design. The NAB style nest box is designed so that people can make a whole nest box from one fence post plank of wood. However, no one has studied whether the nest box depth affects use by bluebirds. Thankfully now, the eastern bluebird population is at a stable place, and there has been research done on the proper hole size to prevent some invasive species from using bluebird nest boxes. There have also been studies done between nest box types, but there is still a need for studies focusing on how nest box depth determines nest box selection by bluebirds. So for my study, I've chosen to focus on this portion of the schematic, which shows the design plans having a depth from the entrance as 6 inches. So the question I'm asking is, does nest box depth affect eastern bluebird nesting success? Eastern bluebirds prefer grassland habitats with few trees and little to no undergrowth. It is due to this reason that bluebird trails are commonly set up in open rural areas such as pasturelands, parks, cemeteries, and golf courses. For this study, we observed 30 nest boxes on Lonnie Pool Golf Course, 15 boxes on the front 9, and 15 boxes on the back 9. For my treatment, I varied the overall depth of the nest boxes by adding 1, 2, 3, or 4 small wooden pallets to act as a false bottom. For the purpose of presenting my data, I ran my statistical test to focus on the comparison between my independent variable, pallet height, to dependent variables, nests produced, and eggs laid. However, I also took more data, which I'm listing as supplemental variables. These are the number of eggs hatched, the number of surviving hatchlings, and the number of dead hatchlings. Here's a better visual of what each series would look like. Each pallet added 1.5 centimeters of height. To save space, I excluded a picture of 0 pallets, but I hope you can visualize what it looks like with the provided images. Because there were 30 nest boxes, there were 6 series total. Each series contained 5 nest boxes, with each box containing an individual treatment of either 0 pallets, 1 pallet, 2 pallet, 3 pallets, or 4 pallets. 4 out of the 5 treatments are shown above, each listed with their respective height added to the NAB-style nest box design. Here are some observation pictures and a segment of my raw data. For the purpose of condensing my analysis of the study for presentation, I focused on nests produced and eggs laid, and used the data from these 2 columns to run my statistical tests. However, I used the supplemental data to calculate percent eggs hatched and percent chicks survived as measures of fitness, because the supplemental data is still valuable to help visualize bluebird reproductive success. Throughout the study, I also listed notes for other intriguing observations, such as a silver-haired bat was found in one of the nest boxes, as well as some wasp nests were found. I also noted when any management activities were taken. This box plot shows how many nests were produced at different heights added. Each bar and box represents the distribution of nest production for that treatment. At first glance, we can see that 0 height added has the highest median, as well as having a high variability. We also see that at an added height of 1.5 centimeters, there weren't many nests produced. However, there is one outlier. 3 centimeters, 4.5 centimeters, and 6 centimeters show some nest production, but are variable and have lower medians than 0 centimeters. This means that bluebirds may prefer the natural height of NAB's nest boxes. There also may be a factor that a slight decrease in depth causes for nest building, but overall, we can take from this that increasing the height of the floor allowed some, but not very reliable nesting. This box displays eggs laid at different heights added. The y-axis shows the number of eggs laid between 0 and 5, and the x-axis shows the different height treatments. From this, we can see that at 0 centimeters, where no height was added, is more successful for egg laying, and any additional height resulted in almost no eggs laid, but there are a few outliers. This suggests that bluebirds may be sensitive to changes in nest box depth, and adding height to the floor likely changed something, whether that be stability, microclimate, or cues they use to decide when a nest box is suitable. After running a Shapiro-Wilks test, I found that both p-values are far below 0.05, so we rejected the null hypothesis that the data is normally distributed. Since the data is not normally distributed, we then used the Kruskal-Wallis test. The Kruskal-Wallis test detected no significant differences between treatments in the number of nests produced or eggs laid based on pallet height, though this may have been affected by some outliers in a few of the nest boxes. So this means that statistically, based on Kruskal-Wallis test results, pallet height does not impact nest production or eggs carried. We next used a chi-square test to determine whether pallet height significantly impacted the presence or absence of nests or eggs. This was done by changing the numerical values of nests produced and eggs laid to be based more on presence. When comparing height to nest presence and egg presence, both p-values are greater than 0.05. Therefore, the chi-square results suggest that pallet height also does not impact the presence or absence of nests or eggs. Some potential confounding variables are the height of the nest cup. A nest cup is used to support nest shape. People often use fruit cups, which are shown in the top picture on the right, or people use store-bought nest cups as seen in the bottom picture on the right. At Lonnie Pool, the nest cup type varied across nest boxes, but most of the nest boxes at Lonnie Pool had fruit cups. I did not include this factor in my data because I believe that the additional height the nest cups would add would be insignificant. A second confounding factor could be small sample size. Having 30 boxes to work with is fantastic. However, 30 is the minimum number needed for statistical studies such as this, so more boxes would be better. A third potential confounding variable would be slight nest box inconsistencies. Although these nest boxes are all the same NABS style nest box design, they may vary ever so slightly due to human error while building them. From this study, I learned that results were not significantly different between treatments. Because the results between Kruskal-Wallis and chi-square both showed that pallet height did not impact nest production or presence of eggs or amount of eggs. However, the statistical significance may differ from graphs due to small sample size. This is good news for bluebirds because it means they can successfully build nests and lay eggs across a variety of nest box structures. When considering this, these results can help inform nest box designers by suggesting optimal depth to ensure that bluebirds will use the nest boxes. However, more research is needed to determine what nest box depth is most optimal for maximizing eastern bluebird reproductive success. And future research could focus on whether nest cup height affects nest production, eggs laid, or eggs hatched. These are my resources. Any questions? Yes.
Great presentation. And
Thank
super
you.
Interesting. I'm just curious, I don't really know that much about birds, but is this the type of thing that could be expanded into some sort of like citizen science project to increase your sample size?
Yeah, definitely. Because this was more like an independent research project I designed, and because I'm working with the North Carolina Bluebird Society, there was another student who was working with the North Carolina Bluebird Society, but she graduated before I sort of started this study, so it was mostly just me and my mentor working on it. But I definitely think it could be a project that could be
more broad to get more hands to collect more data.
My yard.
Yeah.
Really cool. Thank you.
Yeah, thanks for the question.
Did they all make it to adulthood?
Not all of them. No.
No.
Yeah, I did note how many hatched and how many died. I don't know those numbers off the top of my head. But with this slide, you can see that 90% of them survived. So not all of them, but a lot of them. And you had one?
Yeah, so they, in your first box plot, you had a lot of variation, but there were nests built across a lot of different pallet heights, right?
Yeah.
But then your egg laying seems to be the more limiting factor, because once they lay eggs, you have high survival and hatch rates.
Yeah.
So do you have an idea of what other variable might determine whether bluebirds are actually gonna lay eggs?
I'm not particularly sure. I laid the different treatments out numerically, so there wouldn't be like two twos next to each other. So maybe location. Some of the nest boxes on Lonnie Pool are closer to I-40 on like the bottom here. So these ones were more closer to the highway. That might cause some disturbance that might deter them from nesting there. And then like the clubhouse is up here, so there would be more human activity there as well. That's the only thing off the top of my head that I could think of.
When you launch your citizen science project, you can get to the bottom. It's awesome. Any other questions? Thank you, Shea.
Thank you.
Hi, I'm Ben Warfield. I'm a senior in the Fisheries, Wildlife, and Conservation Biology program. And for my minor research, I did a herpetology inventory of Lake Raleigh Woods. This is a 100-acre almost urban forest owned by NC State University. This is on our Centennial campus. So if we see this sort of up here area, this is where all of our class buildings and the library is. So my side is on the other side of the lake. This is a really, really cool site. There are 12 recognized community types, and it actually has a relatively complex topography for being an urban forest in the Piedmont. It's got pretty steep slopes at the back here, streaking down into the lake. It's very cool. This is a hallowed space on NC State. So that doesn't really mean anything in terms of legal protections, but it does mean that NC State owns it right now and is not interested in selling for the time being. Again, there are no legal protections on it. It's a really, really cool spot. It's really diverse because of those community types, but it is still under threat. So primarily due to invasive species, there's a lot of Eleagnus umbellata and a host of other things. It's also pretty polluted, as you can imagine, being a lake that's on a college campus. So for study methods, not super strict. I basically just took long walks out in the woods as much as I could. I did use cover boards, which are basically just wooden boards that you throw somewhere and then hope you find something under it later. And then kayaks. I used the kayak to try to paddle around some of the aquatic vegetation in the islands, see if there was anything on the places I couldn't reach by foot. I went out almost weekly for the entire year. I spent a little over 100 hours out here. So to talk a little bit about what we thought we would find. So this is a map of the site to kind of walk through some of the habitat types to get a little bit of a better view of what we actually see when we walk through these forests. So I'll go ahead and start down here. Communities 4, 5, 6, and 7. These are all pretty much wastelands in terms of herps. There's almost nothing. It's a pine successional forest, so there's no downwoody debris. There's no herbaceous layer. There's just nothing really there for any herps to use. As we come up, we'll talk next about the mesic areas here. So these are community types 9, 11, and 12. And so these are more of our wet areas. They're kind of divided into two parts. So a little bit up the hill, about right here, we have rocky streams, and these are streams with rocky bottoms. They're very shallow, but they have moving water, which is significant. And here we think we would find our 2- and 3-line salamanders, a lot of our Eurecia genus. And then as it comes down the slope, it opens up about here into these really wide, boggy, flat bottoms. And these are characterized by having really mucky soils, a lot of standing water, a lot of mud, a lot of silt, just really swampy areas. And this is, in the boggy, flat bottoms is where we expect to find really most of our amphibians. So we expected to find a lot of our dusky salamanders. A lot of our salamanders are Chamberlain's dwarf salamanders. A ton of cricket frogs. We expected to find a lot of cricket frogs. All of our lithobates species of frogs, which is our pickerel frog and our green frog and our southern leopard frog. And lastly, kind of our Anaxaris, which is our American and Ballard toads. So moving up into community type 8, these are the mesic mixed hardwood forests. So these are kind of an intermediary between our lowest, most mesic areas and our more dry and upland sites. So they kind of have rocky soils, but they're really next to a lot of the wet sites, a lot of things like that. So here we expect to start finding our slimy salamanders, which are a little bit more upland than our other salamanders, and then start seeing skinks, our first reptile, and again, our Anaxaris toads. Then finally, moving over to community type 2 and 3, these are dry oak hickory forests. So these are our highest and driest sites. They're going to have really rocky soils, a good bit of dead, downed woody debris, and a pretty consistent herbaceous layer on this site. And here we expect to find our small fossorial snakes, like our Decay's brown snake and our worm snake. We expect to find more slimy salamanders. These are our woodland salamanders, and fence lizards and anoles rather than skinks, because they're neighborhoods and other buildings along this side of the site, so we get a little bit more human structure for those lizards to use. So moving on to what we actually found. Start with the amphibians, because this is probably the most interesting part of the survey. For the most part, everything followed what we thought we would find. In a spurt of what I will call optimism, for lack of a better word, we thought we could find spotted salamanders up at kind of the top left area of the site, because that is where Lake Johnson feeds into Lake Raleigh, and there are a lot of streams and islands in that kind of area. And so we thought there could be a consistent amount of ephemeral pools that would serve as breeding habitat for spotted salamanders. We went out there, didn't find any spotted salamanders, which makes sense because we actually didn't even find really ephemeral pools. So there was no breeding habitat for them, just characterized by having ephemeral pools that are serve as refugia from fish so they can't eat their eggs. So no spotted salamanders, not totally unexpected. The next kind of weird thing is that we didn't find any 3-line salamanders the entire year, which is kind of odd because the habitat requirements between 3 lines and 2 lines are so similar. I would imagine that if you would find one, you'd probably find the other. But in my year of searching, I did not find any. Couple more things. This guy was probably the most intriguing aspect of the survey. That is an eastern newt. I didn't really expect to find him. And I found one, and I found that one several times throughout the year. So right about there's a group of about 3 or 4 logs that are like a foot or 2 off the trail. And week after week, every time I came out, it would be that salamander under one of those 3 logs. I have no idea why. That's not really where he should be. If he was going to be anywhere, he should be in the lake, honestly, but closer down to more of the boggy flat bottoms. Every time I was out there, he looked about a day from death, but every time he was still there under those logs. It was pretty funny. And the last thing to note for amphibians is that the frogs are probably a little underrepresented in terms of the habitats they actually take up, because a lot of the times I got frog records, it was from their calls. And you can't tell where they are when they're making that call, since there are a ton of them. So this is a Cope's gray tree frog right here. That was probably the biggest kind of example of that effect, because I heard a lot of them throughout the entire year, but I only got hands on one, and he was up in community type 3 where he shouldn't have been, honestly. So it's interesting. So moving on to the reptiles, where we found a little bit less, or I guess less than we thought we would. Starting with the turtles, again, nothing really unexpected. They're just kind of doing what they do, hanging out in the lake. We didn't see anything too surprising with them. The lizards followed about the same trend we thought they would, with more skinks being found in type 8 and more anoles and fence lizards being found closer to the human structure. But nothing too crazy with the reptiles, except for the snakes, which was far and away the most disappointing part of the year. We only found 4 species and only 6 individuals the entire year at this site, which is kind of crazy. Found 3 worm snakes, 2 Decay's brown snakes, 1 tiny rat snake like a month and a half ago, and then a water snake I saw out in the lake. So not a lot of snakes out there, which is definitely kind of disappointing for sure. And we would have expected more. So kind of a summary of kind of what we found and comparing it to some other things. So I found 26 species total on the site this year. I found 13 amphibians and 13 reptiles. I have the potential for 16 and potential for 24. That's in reference to the general management plan for Lake Raleigh that was developed in the early 2000s. As a part of that management plan, they carried out a habitat assessment basically to see what elements were there that could potentially support this many species. 40 species on this site would have been bonkers. Would have been a really, really crazy number. So I'm definitely not disappointed to not get that. 26 is definitely a good number. So another interesting thing to mention is that on the other side of the lake, about up here and on the other side, that is a greenway, and down right there is a dam for the lake, and following that dam is a rocky creek. And I searched there this year too. I can't count them as species I found in my site because they're outside of the bounds, but I did find a lot more stuff over there, specifically the snakes that we thought we would find on this side of the lake. So we did find one more amphibian as well, a narrow-mouthed toad, which is very, very cool. But we also found a black racer, a ringneck snake, a copperhead, and a rough green snake, all of which we thought were possibilities to find on the other side of the lake. So it is comforting, I guess, to know that they are still there. They're just not in the exact area that I was looking at. I also compared what I found to the iNaturalist records for that site. And they're pretty similar, but also not, which is interesting. So I found 26 species this year, and there are 23 species recorded in iNaturalist. So I have only a couple more species, but they have 4 species that I didn't find, which is interesting. So they have primarily those 3 snakes that we thought we would see on my site that I didn't see this year. They are recorded there on iNaturalist and across the lake. So I don't know what was going on this year, but nothing good. So this work is important for a lot of reasons. So I'm going to start from the bottom, actually. The first, a little bit cliche, but you never know what you might find. So this is actually an albino white-spotted slimy salamander that I found in May. This is the first documented case of albinism in this species, which is really cool. It's kind of a double-edged sword, because if you find an albino salamander in March, you know, the rest of your year is kind of downhill, right? That's kind of the peak. But so doing herps acts as a really good indicator of ecosystem health. So if you have an area where you think you might see a lot of herps, you should have diverse species, you should have an abundance of herps. And they're not there. That's kind of like a canary in a coal mine, that things might not be as healthy as that ecosystem appears, and kind of a cursory look. In that same vein, inventories like this can kind of inform on management. So there is management plans. There is a management plan being developed for this site, which is really exciting. It's really great. And something that I found in this survey is that we talked a little bit about Elaeagnus umbellata, right? The invasive species that that shrub basically dominates a lot of the slopes leading down into the streams. And I found almost nothing on those slopes the entire year. That was far and away, other than the pine stands, that was the least diverse area that we looked at the entire year, was those slopes that were dominated by those invasive plants, because those shrubs crowded out everything else, shaded out any kind of plant that could grow up that our native herbs actually like to use. So it is good to know that by managing invasive plants, you manage a lot more than just the species you're removing, right? So this is trickling down all the way to the herbs that use those streams, because the shade and the vegetation regimes aren't what they want. So, and lastly, the citizen science, it's very helpful. iNaturalist is a great tool, but it definitely can't replace an actual survey look at it, right? It can inform what you want to look at. It can help you examine trends. It's very helpful. So it's really cool that we found a squirrel tree frog on INAP, which is cool, but 3 species that I was like, they should be here. Why are they not here? We know they are there based on the record. So that can tell us if populations are declining or if things, I don't know. It can give you inferences where you might not have data otherwise, but it can't really compare to an actual research drive, right? So it's to be used as a tool and not a replacement, right? Yeah, and so for final words, really like to give a truly huge and heartfelt thank you to Dr. Steph Jeffries and Thomas Reed, who were my advisors on this project, and wouldn't have done the project without them, and it definitely wouldn't have been as interesting without them.
So you mentioned that the autumn olive caused a lot of shade on the slopes. Do you think that increased shade could help to explain why some of the amphibians were further upslope than they normally would be?
I wouldn't say so. So I'll go back to the map. So the slopes that autumn olive dominates are kind of right along here. It's like basically shrouded over that stream. And a lot of the amphibians we thought we would find are more here where the autumn olive hasn't reached yet.
Mhm.
So I wouldn't quite think so, but you never know, right?
Great presentation. I was wondering, in terms of the new management plan, is that something that like you and like your lab, I guess, are gonna be working like with those people in terms of informing? Because it sounds like, just based on what you said, maybe a lot of those species are not being found, or like less of them due to, you know, invasive species, but perhaps just like the pollution that you mentioned in terms of like the lake and things like that. Yeah, so are you going to be involved in any of that?
I don't, not directly, at least. I would like to submit what I found, if possible, because I think something that could explain. So the main thing I was talking about was that 3-line salamander not being there, right? And the habitats that utilizes were the ones that were being directly affected by that Elaeagnus. And we didn't find as many 2-lines as I frankly thought we would. So that was the most heavily impacted part of the site, were those rocky streams, and those were the areas where we didn't find as much stuff as we thought we should have.
Did you see a change in the earth content before they cut down those trees?
At the Chancellor's house? Yeah, there was. So it's kind of weird because the one decayed brown snake I found this year was found directly following that cut. And it was on the side of the cut following that cut, and a log that I have surveyed pretty much every single time I've gone out there. Very familiar with that log, and I've never seen a snake under it until that cut. So superficially, that seems like, oh yes, this cut would probably help herps, right? This would probably do this, because if we found this cut and we find a snake right after, then your first thought is that cut helped that snake, right? But I think what's more likely is that that snake had a burrow or something that was under the affected area, that when those machines came in and they drug up all the soil and they drug up the ferns and the shrubs and everything that was there, I think it probably destroyed whatever refugia it was hiding under. And I never found that snake again, right? So it either left, it left to go find a new place to live, right? And I don't know if it made it, but yeah, anyways, so superficially, it seems like it benefits it, but I don't think that's the case.
Thank you, Ben.
Hi everyone, my name is Taylor, and today I'm excited to share how microclimates can dramatically change how soil breaks down. So I'm going to walk through an experiment that I did using the teabag index to compare decomposition in sun versus shaded plots, and show how this idea can be turned into an outdoor learning experience for students. So this is one of the most important microclimate differences is sun versus shade. These two conditions can create totally different atmospheres, including temperature, moisture levels, and decomposition rates just a few feet away from each other. So the goal of this study was to compare how these microclimates affect how fast organic matter breaks down, and then think about how this could be a simple, engaging ecology lesson for high school students. So this is the courtyard where the study took place at Athens Drive Magnet High School. This is actually where I attended high school, and my mentor, Angela Clark, is a teacher at this school. So currently this space is really never used, especially for anything academic or scientific. She said that she only sees students vaping out there. So hopefully we can make this a lab instead of that. And then this is just a better look at the inside of the courtyard. As you can see, it's a really big space, and it's a good confined area, I think, for students to be able to use for like experiments. So a microclimate is basically a mini climate created by small environmental differences. Things like sun exposure, nearby buildings, and vegetation can make one patch of soil noticeably warmer or more moist than another patch just a few meters away. And these differences may not seem huge, but they can strongly influence the decomposition and soil health of an area. So to measure decomposition consistently, I used the teabag index. This is an inexpensive, standardized method used worldwide, and the green tea is meant to break down quickly and helps calculate early stage decomposition, while the rooibos tea breaks down more slowly and represents long-term carbon stability. So this is perfect for schools because it's affordable, it's easy to follow, and the procedures use real scientific data. So I tested two hypotheses. My first was testing if sunlight would lead to warmer soil and if that warmth would increase decomposition. And I hypothesized that it would be increased in the sun because I did this in the colder season. So I figured the warmth would have and temperature would have a bigger effect than moisture in the colder seasons, just based off my research. And my second hypothesis, based on the factors of the teabag index, was that the green tea would decompose faster than the rooibos tea because it contains less lichen, and so it's more easily broken down by the microbes. So I set up two plots, 2 in the sun and 2 in the shade, and each plot received 5 green tea bags and 5 rooibos tea bags for a total of 40 samples. And as you can see there, I use these like little sticks to keep where the bags were in the experiment. So the bags were buried at consistent depths. After the incubation period of about 7 weeks, I dug them back up, cleaned them, dried them out in the sun, and recorded their mass loss. And this setup mirrors the official teabag index protocol, so I could ensure that my results could be compared to global data. And then here you can see the actual plots in the courtyard. That's the sun plot, and then this is the shaded plots. Oh, sorry. That's a better picture of them. So before burying the tea bags, I collected environmental data for each plot. I took the soil temperature, the air temperature, and the percent canopy cover. And these factors help explain the differences in decomposition. And soil temperature specifically is especially important because microbial activity increases with warmth. So here you can see the actual microclimate differences that I recorded. The sun plots had warmer soils at about 65, and then the shaded plots I recorded at about 56 degrees Fahrenheit. And that's what I expected to speed up the decomposition. So the shade plots had a higher canopy cover as well, which reduces the sunlight and tends to keep the soil cooler and more moist. I wish I got better pictures, this was when I collected my samples, this was the canopy cover, and it was definitely more apparent. And I wish I got better canopy cover pictures when I planted the samples, but it was definitely a significant difference between the sun and shaded plots. So this was my method. I just buried the tea bags and then I waited 7 weeks, retrieved them, dried, weighed, and calculated my variables. And here's some examples of what the tea bags looked like. And I was only able to recover 34 of the 40 bags. But this is pretty normal for outdoor experiments because of animals, water movement, soil shifts. But the 34 bags was still able to give me a good amount of data. So here are my results. For both the green tea and rooibos tea, the mass loss was greater in the sun plots, which supported hypothesis 1, but it wasn't a significant difference, as you can see. I think this was largely due to the temperature. In colder weather, it's a lot slower decomposition rates. And also the incubation time wasn't a full 90 days. It was closer to 50 days. So I don't think I got as extreme differences as I might have seen in a warmer climate or in a longer incubation time. And then this one shows better how directly the green tea consistently lost more than the rooibos tea, which supported my second hypothesis. And that makes sense because the green tea was supposed to decompose faster based on the teabag index. So it's good that we see that consistently across the results. So the S is the stabilization factor, which is based on the green tea. And that factor, it reflects the long-term carbon storage, while the K represents the early stage decomposition rate, and that one's based off the Rubio's T. And these values let us compare the courtyard to global ecosystem using the T-bag index. So the S value of 0.33 is actually pretty consistent with the research studies that I compared my study to. And that's the mean stabilization factor found in large-scale citizen science projects that I observed. And this S value is relatively high, but the K value is low at 0.02. And this signifies that the decomposition rate was very slow. And like I said, I think that's due to the colder weather. So that means not a ton of organic matter was decomposed, which makes sense with the incubation time and weather. So like I mentioned, I wasn't able to find every single tea bag, and in the colder months, slower decomposition. I will say my mentor, Angela, we have goats at Athens Drive, we have animals. So they let the goats into the courtyard to clean up some debris, like eat all the excess leaves and everything. And one of the goats did pass away in there. So I'm not sure. I don't think they stole the tea bags, but I don't know what happened in there. And so beyond the science, this project shows how an underused courtyard can become a hands-on educational space for students who can monitor soil, design experiments, and contribute to data and global science projects, which I think is really important for high school students, especially with this being a magnet high school. And it's also important to develop a sense of stewardship for the school and the environment. I know when I was at Athens, I used the courtyard to do like a cleanup project. And so I'd love to see other students using that space for science and other things like that. So yes, the T-bag index is a part of a worldwide citizen science initiative, and anyone can upload their results to the map and compare decomposition across ecosystems. And the protocol is super simple. I put it here so students could easily follow it, and it's simple enough and cheap enough for a high school to implement. So this project shows how Courtyard can function as a real experimental space, even with a simple method like the T-bag index, because the courtyard already contains strong microclimate contrasts like sun versus shade, warm versus cool, dry versus moist patches. Students can generate meaningful ecological data without specialized equipment. And I think that's really important because repeating this study across seasons or classes would let students measure how climate shapes decomposition, nutrient cycling, and carbon turnover, and essentially transforms an overlooked outdoor space on campus into an applied ecology lab. So looking ahead, I think that this project opens the door to a wide range of student-driven opportunities. Like I discussed, they could just monitor different stewardship projects or other types of experiments in that space. And I'd like to thank my mentor, Angela Clark, for helping me with this project and letting me use that space at my school.
Do you think your decomposition value was low because you let it run for 50 days compared to 90, or did you scale it?
I think it's because it was for a shorter amount of time and also just with the colder weather, but at least the green tea, I could see that it was working in that time. So I would have liked to kept them in there for a long time, but Ms. Clark said she's going to work with the STEM coordinator and have students doing this. So I'm excited to see their results in different seasons.
It's awesome. And great to see that those boxes got rebuilt because they were struggling for a little bit.
Yes. Thank you. Oh, yes.
You had sense that it was unusually cold during that period, time of temperature rates compared to what you would normally expect?
I'm just in the fall semester versus I think if I had done this in the spring semester, I think I definitely would have seen warmer climate and like more decomposition. But like I said, it also could have been the incubation time.
All right. Hi, everyone. My name is Emy Tran, and for the past about 2 semesters, I've been working with Dr. Hannah Levinson and Dr. Bradley Metz to assess the impacts of project designs on pollinator research. And so my research continues, ongoing research by Dr. Levinson and Dr. Metz, hoping to measure the impact of different sampling methods on the overall amount of individuals and different species collected, as well as the impacts of sampling and collection methods on population results within the field of entomology and more broadly within the field of arthropods and beyond this, hopefully within the collection of wildlife for conservation reasons. So our overall scientific question is, how do differing sampling methods and habitat types impact the species richness and abundance of pollinating insect communities within a given ecosystem? It's important to note that Dr. Levinson's initial study did also include a preliminary literature review, culminating in a published meta-analysis, which you can see here. I recommend reading it if you have some time. It's a good time. But this initial review was slightly limited in that it only looked at agricultural habitats, or within the meta-analysis, they're referred to as agroecosystems. And so I was brought on as an undergraduate research assistant in hopes of helping to expand these findings and apply them into other habitat types. And so I was brought on specifically to expand these into forest habitat types. My goal is to do this through conducting a similar, very vast literature review in hopes of also creating a meta-analysis looking specifically at insect populations and sampling methods in forest ecosystems. And so to do this, we conducted a new study with a new scientific question, which assessed how do differing sampling methods impact species richness and abundance of bee communities within forest ecosystems. And so, like I mentioned, I did a literature review. We got our pool of our initial 139 relevant scholarly articles just through a library pull using a 2-criteria methodology. And so these 2 criteria both included lists of key terms, which were used in tandem. And so our first criteria looked at our species of concern, and it included key terms like pollination, pollinator, bees, native bee, wild bee. And then our second criteria focused more on our habitat type. And so it included key terms like landscape, habitat, semi-natural, natural, floral. Once we had all 139 of those articles, they all went through our preliminary screening stage, which is what I like to lovingly refer to as the how did you even get in here stage. Because surprisingly enough, this is where most of our articles were bumped out. This primary screening stage seeks to answer 3 big questions. The first is going to be, is this primary research? So can this even be used in a meta-analysis? Does this research look at more than one group of pollinators? So not singularly honeybees, but also wild bees, native bees, non-bee pollinating insects. And then our final is, was it conducted in a forest ecosystem? And so this final question is really what made it tricky for us somehow, because many judgment calls had to be made regarding what a forest ecosystem was, because there's not really much of any kind of standardization across these studies. So according to the National Science Foundation, a forest is a biotic community dominated by trees and containing a complex interconnected web of plant, animal, microbial, and fungal species. That could be a lot of things. That's pretty broad. And so I chose to really focus on that complex interconnected part. And so I chose not to include anything labeled as a shrubby area, a woodland, agricultural forest, or an orchard that contained a majority of a monoculture, just because I felt that the ecological makeup of those kinds of habitats were going to be too different than your traditional forest ecosystem. And so once we had a yes to all 3 of those initial questions, we were able to move on to our secondary screening stage, which again, articles could get booted out from this stage. But this looks to see, is our study talking about species richness and species abundance, what our study wants to know. And so again, we needed yes to all of these questions in order for our article to be moved on to its final screening stage, which is the extra information. So we're looking at logistic details like where was the study conducted, how long did the study last, when was it started, what sampling methods and collection methods were used, the number of sites, number of visits to sites, and then more specifically, like I just mentioned, were species abundance and species richness mentioned. But beyond that, were they mentioned individually by collection method type and then cumulatively overall? And so after all of this screening, we ended up with a whopping 8 relevant sources. Definitely not as robust as we were hoping for, but we continued on with our research and it did our statistical analysis using the data from these 8 relevant sources and compared them to the data from the initial findings from the agroecosystem study. And so here we have a little bit of the results and some of the breakdown. So here you can see our study duration. The biggest takeaway from this is going to be our negative correlation between the start of the study year down here and then our study duration, so the number of years that the study took place, our y-axis. That negative correlation essentially just means that the more recent a study started, the less time the study has been conducted for, which is what we would expect to see, because if something began in 2024, there's only a year for it to be conducted, whereas if something started in 2000, which we did have a study, I want to say, that started in like 2008 and went on for 15 years or something like that. So this is definitely a correlation that is significant. But even more than that, the forest results did generally seem to be pretty similar to our findings from our agroecosystem studies, or within each of these figures, those agroecosystem results are going to be referred to as field results. Another important takeaway is that our median study duration was about 2 years. Again, this is something that we are going to not be surprised to see, as that's pretty comparable to a typical master's program, which is what a lot of these studies are going to be conducted through. Next, we have our sampling sites. Our sampling sites are pretty broad, both in the field and forest ecosystem studies, and overall our forest ecosystems were, again, pretty comparable to our field. Here's where you can see our forest studies kind of shining in their own light. So our field studies tended to have fewer sampling trips per year. They're going out usually maximum around 10 to 12 times per year, once a month. Alternatively, our forest studies, even with our very limited sampling size, you can see those sampling trips tended to be much more frequently throughout the year. And that is significant because we also noted a correlation between the higher number of sampling trips and the use of traps as sampling methodologies, which again would be something that we would expect to see, just because it's going to be a lot easier for researchers to go out, check those traps, maintain those traps multiple times in a month, as compared to going out and sitting in the field and observing these species for multiple hours, multiple times a month per year. Here you can see a little bit of a deeper breakdown into the sampling methods that we used. So over here you can see our collection methods that included trapping tended to have higher sampling time. And again, something that we would expect to see for similar reasons that I just mentioned. It's going to be a lot easier for researchers to go out, place those traps in the field, leave them for multiple days. Whereas if they're going out with a net to catch insects, they're not going to be able to be out in that field for multiple days catching insects. Another important thing to note, again, is that our forest results were generally pretty similar to our agroecosystem results. Next, we have our species accumulation curve. And our largest takeaway here is that there's a positive correlation between species abundance and species richness. Again, something we would expect to see. This essentially just means that the more individual insects that were collected in the field, the more distinct species that were able to be identified. And you can kind of see that with the exponential growth curve here. Our exponential growth curve, with the statistical analysis that we did, that model indicates to us that our sampling saturation is going to be at about 10,000 individuals. What that means is that you can expect to reach the maximum number of distinct species within your ecosystem, habitat, whatever it may be, once you have sampled 10,000 distinct individual insects. And that is true both in the field and forest studies, which is pretty great to see from our very limited sample size, that again, our forest results are generally pretty similar to our field findings. Here we have our species richness abundance residuals. And so here it's very important to note that our residual richness model was trained on the initial data from our agroecosystem or our field study. And so essentially what that means is that residual richness is determined based off of a model that is created by the statistical data, essentially. And so these models are created in order to predict what your richness should be for your habitat. So it's kind of like your ideal richness. That ideal richness is then compared to the actual true richness that you found in your study, and what you're left with is your residual richness. And so our residual richness was completed only using the data that was found using the agroecosystems meta-analysis initially. And so our figure shows here that the richness of the forest studies were frequently underestimated by the model that was trained on our agroecosystem data. And so basically all of that just means we can infer that our forest ecosystems are going to be much more species rich than our agroecosystems or our field data. And so all of this information and all of these statistical analyses have been conducted in hopes of creating the Levinson Index. And the Levinson Index is an application that is hoped to be used by land managers and researchers and anyone in between who wants to be able to predict the species abundance or species richness of insects within their area. And that'll make a little bit more sense in just a second. So here we have a sample of what the Levinson Index looks like. And so your area could be a field if you're a farmer, it could be your plot of land if you're a land manager, or it could be whatever your study is if you are a researcher conducting a study. But you can go ahead and fill in your study design. And so over here, our individual on the right has 10 sites. He is studying for 3 years. He's going out to spend 100 minutes. He's going to go out once a month, so 12 sampling trips a year. He wants to identify his insects to genus, and he is going to sample them by visually observing them. And so our Levinson Index predicts that he should expect to find roughly 250 distinct species after he's collected about 6,000 individuals. And so it's important to note that the Levinson Index, while it is great, it is limited also to the model that it was trained on, and that is the data that was collected using the agricultural meta-analysis. And so our next steps are hopefully to expand, which is why I was brought on, expanding into the forest ecosystem world. But even more meta-analyses for future students in ecology. There's a lot of potential growth for them to do similar meta-analyses for ecosystems of all types. It is very important to note, like I mentioned, my pool was very, very small. I ended up only with 8 relevant articles. And while our results were very promising based off of those 8 articles, unfortunately, that is just not how science works. So hopefully we can get students in the future coming into a project similar to this, conducting another meta-analysis using forest data similar to the one I just did. But I would recommend altering those key term criteria lists to be much more forest-centric and forest-focused. And then again, I would recommend doing even more meta-analyses to further broaden the Levinson Index into different kinds of ecosystems as well. That's it.
You know I have a question. So you screened 139 articles.
Mm-hmm.
Please tell me that you have information about like the shrubby areas, the woodland and orchard, because don't you think like you could go back through and compare your 8 forests to those different, like they're not agroecology systems, but they are maybe more comparable to forest?
So we had a lot of articles that were knocked out because they were orchards specifically or agroforests or something of the sort. And yeah, all of them were labeled or written out and described specifically what it was. But the way that each of the screening stages worked was that
knocked out at multiple levels.
Yeah, so when it would get knocked out on the first one, I would still have pretty much all of the rest of the information filled out. So here you can kind of see this is the information that you were looking for all of the articles. So when I would go through and screen these articles, I might not immediately know whether or not it looks at more than one group of pollinators. And so a lot of the time before I would even determine if it was knocked out from our primary screening stage, I would have a lot of background information in like this area of screening. So yeah, a lot of that information is there. And throughout the screening stage, we also had like a different sheets that was other ecosystems for future research. So there's definitely a lot of extra information that would be a great next step research project.
Thanks.
Great stuff, Emy. So why do you think studies in forests, they're able to go out 35 times or whatever, right? Compared to agroecosystems. Do you think it's because there are like forestry people actually living on the land? And could that also explain why you also only were able to find 8, right? Because there's not very many people living in cabins in the woods doing these kind of things.
Yeah, I definitely think it's very people like focused. I think it's a lot easier to go out into an agricultural field just in general and net for something or observe something. There's going to be a lot less distinct hiding places for insects, I think, in the monocultural area. But also the agreements that they have with the landowners for the agricultural study, like it's a lot more likely that we have an agreement that, yeah, I can come out to your farm and do what I need to do once a month rather than whenever I feel like it. Whereas these forest ecosystems, I feel like a lot of the researchers have kind of agreements with sections of forests or land managers in the forest where it's, you know, I'm doing this to conserve your area. It's going to mutually benefit us both. So I'm going to be able to make my study as beneficial for me and as accurate as possible without having to work through the hoops of you're letting me use your land because you want to type of deal. Yeah.
Yeah, very big meta-analysis. Thinking about the ones and some. I think the excess of species in some for the very samples that they took, do they sample to a greater range of species?
I do very much only from the sample size that we have. I would say I do, because of the analysis that we did indicates that we should be collecting about the same. We should be getting about the same results of species richness in comparison to our abundance as we are in the agricultural systems. So I feel like from our analysis, that is kind of the only conclusion that we can come to, is that our increased richness in the forest ecosystem is going to be from increased sampling. From my personal perspective, also doing the analysis, I would say something similar. I noticed a lot of the forest studies go on for a lot longer, and therefore are sampling not only more frequently, like we saw, but for longer durations. And so they do have a much more vast collection of insects and distinct species. From an ecological view, I would also just say it's likely that there are more diverse insect species in a forest ecosystem, especially an undisturbed forest ecosystem or an old growth forest ecosystem, like a couple of these were, as opposed to a lot of the monocultures and croplands that they are going to see in the agricultural study, just because of the complexity of the landscape.
Hi everybody, my name is Kaleb Holder, and today I'll be talking about an age-old question. Does size really matter? But in the context of bees. So we're looking at if body metrics and body condition, how they affect sperm viability and sperm counts in non-model bee species. I really want to start by saying thank you to my mentors. Luckily, this has been a collaborative study between the Dr. Irwin's lab and Dr. David Tarpey's lab. But I really want to give a special thanks to April Sharp and Dr. Brad Metz for just the countless hours of mentorship and really asking all the strange questions that had came to mind throughout this study, in addition to offering the lab space and really trusting us with the overseeing, especially of the Queen Center. So, what is a model species? This is a highly debated topic, but in the bee system, we're going to be saying that the model species is honeybees for this. So our non-model species are 5 that we have selected are Xylocopa virginica, or carpenter bee. We have the Osmia cornifrons, Osmia lignaria, Bombus impatiens, as well as Megachile rotundata. In the Irwin lab, we focused specifically, with the April, we had been focusing with bombus impatiens or bumblebees. And throughout the study, we observed that, we were only focusing on females in this study and that the males often were discarded or culled. And so this opportunity arose to make use of the bees that were previously going to waste in a sense. So we wanted to give their life more purpose. And so following a conversation with the TARPy lab between April Sharp and Bradley Metz, they came up with this idea of trying to establish baseline sperm counts as well as answering some questions in regards to body metrics and body condition. So we wanted to establish some methods because in literature, there's barely any research regarding these bee species in terms of baseline sperm counts, viability, as well as any methodology. Really, the only data that's in existence right now, I think, is 2 papers on Osmia cornifrons. And so we kind of based some of our study methods based on those. So our question, of course, is which impacts more, the body size metrics or the body condition on sperm viability and sperm counts? So how did we conduct these studies? Here we have the body condition. We are looking at things like thorax weight, body weight, and age. And for body measurements, the intertegular distance and interocular distances. Our predictions was that body condition has a higher effect on the sperm viability and sperm counts for both bees. The way that we did this was we sedated the bees using carbon dioxide gas. We then extracted their seminal vesicles and macerated them into a sperm buffer, and then did manual counts on a hemocytometer. In terms of taking intertegular and interocular distances, we used ImageJ to measure the distance from the furthest on the outside of the eyes of the bee, as well as the joints for the wings. For the statistical test that we ran a principal components analysis to observe any sort of trends that we could then go forward with investigating, as well as a linear regression Here we can see that the OCD, so the interocular distance, and ITD, intertegular distance, were both clumping. So showing that there was a correlation between those 2 variables, in addition with the other body condition measurements as well. So in terms of Osmia lignaria, these linear regression data is just presenting the raw data, but we do see a positive trend between the thorax weight and the manual sperm counts. In addition, we're seeing a positive trend or a positive relationship between the manual sperm counts and the average sperm viabilities. In continuation of this, we're also seeing that the thorax mass does have a positive relationship with sperm viability as well. Across the majority of these species, we're seeing that thorax is one of the main drivers in sperm viability and sperm counts. In Osmia cornifrons, this was the one species that we found that as they are getting older, their sperm counts are decreasing. But the body mass has a impact on the sperm count in a positive relationship between these. In terms of Megachile rotundata, we weren't able to find any significant data yet, but we have a theory that it's potentially because in species like Osmia lignaria that over winter, they have to have a larger store of energy that tends to get used up throughout the winter, whereas Megachile rotundata, they are maturing up until the point that they emerge from their cocoons. So we think that they might be more resistant to changes in age. Okay. For bombus in patients, we're seeing an increase or a positive relationship between sperm viability and sperm count, as well as a positive relationship between thorax mass and between the manual sperm counts. This again was continuation of the trend that the thorax mass is impacting the sperm counts and viability between the 2. We see a reverse though in the Xylocopa virginica. where we're seeing that the body mass is there's a negative relationship between body mass and the manual sperm counts, and also a slight positive trend between average sperm viability and average sperm counts. There is a bit of April and I have been discussing whether the linear regression for this was really the best thing to use, because our linear regression is not going to the full extent of the graph. So it's something that we want to expand further on in the future. So answering this question, does size matter? Luckily, it doesn't seem that size is the main driver of sperm counts and sperm viability, but rather the body condition. So what we're seeing here is that I apologize. The limitations here is that the Xylocopa, we were collecting those wild, so we weren't able to see how old they truly were, nor if they had mated previously, which will affect their sperm counts. In addition, there was a bit more methodology that went on behind the scenes of this study. We have been using a cellometer to take the sperm viability as well as sperm counts, but we need to optimize the cellometer just a bit more because we're finding that it's counting the non-sperm cells, and so we want to make sure that our counts are as accurate as possible. So the most reliable way was to use our manual counts for this study. We were hoping and we were curious to see if there were going to be any trade-offs between the sperm counts and sperm viability depending on life history, but we are going to have to wait just a small bit longer to figure out to answer this question. We are really curious about the Xylocopa just because we are seeing that negative trend or that negative relationship between the body mass and sperm counts. So I think that would be a really interesting place to start potentially if we can get a good supply of the Xylocopa. But yeah, the next steps again is going to be optimizing those cellometer counts, because once we do that, we're going to be able to run these tests a bit more reliably in the future. But yeah, I want to say thank you again to my mentors. Again, it was so much work that went into doing this, and I know that, I just really appreciate y'all's help. I know that I could reach out to y'all at any time, so I really appreciate that. And special thanks to Dr. Irwin and April Sharp, again, for really taking me under their wing. And again, thank you to y'all for being here for the presentations. I really do appreciate y'all's presence. I know that there's just a lot of information, and thank you all again so much for sitting through it. Yes.
Besides bees being cute, that's all I know about them, really, and that they're good pollinators. So in terms of sort of wanting to understand more about sperm count and viability and things like that, so what would be, knowing that information, like how does that apply to real world conservation initiatives? I'm very, very happy that you asked this because it had slipped my mind when I was presenting. But what we're seeing is a trend among animal species is as the increase in anthropogenic activity is going on, we're seeing a decrease in sperm counts and viability. So if right now we're only able to rely on honeybees, it doesn't really give us a big whole sense of what's going on within the ecosystem. So we're hoping that by promoting other model species to analyze environmental health, we can have a better understanding of what's going on within the ecosystem. If we're seeing that across all, in this example, 5 species, we're having a decrease in sperm counts, then what's going on in the ecosystem that is influencing that?
Why did you choose to measure thorax size rather than size?
So the thorax size, based on literature, this is to the best of my knowledge. I don't have the literature to pull from exactly, but to the best of my knowledge, the thorax is one of the more reliable measures between different species. So you can kind of compare that way. Yes.
So you said body condition seems to be a more consistent, reliable predictor of sperm count and viability than body size, right? I guess in a world where everything is changing, right? Does that make you more comfortable than if it were size that might, to me, it seems like condition is more promising as long as they can find sufficient resources across the landscape, whereas body size might be affected by changing temperatures and things. But
Mhm.
but that's me and I'm no bee expert. So do you feel more comfortable or, more nervous about condition versus size?
This is speculation again, but just based on what I can predict on my own, not based off of any like sound statement. I am unsure because, as the changes are going on within the environment, it changes differently. And so we haven't been able to test the hypothesis that, is it that being overwintering in a cocoon is really what's causing the decrease in the fitness or the body condition. And that's kind of what is allowing the osmium cornifrons to we suspect have a higher fat content. So I think there's going to be need for more research around that to kind of see how different environmental factors will impact different species. Yeah, I think it's a very specific context dependent question.
Okay. Thank you, Kaleb. And thank you all so much for coming out today in our, beginning of exam periods at NC State. Really, really appreciate our students supporting each other. Really appreciate our students who presented today. So congratulations to you all on your fantastic work. Thanks so much.